will warn y'all. Some of y'all's back of your heads are in the recording now, by the way. All right. So we're going to start our new topic, which is viruses. So unfortunately in the book it kind of introduces it and tells you how to grow them, which we don't need to know for this class, which is great. So we're only going to cover parts of Chapter 5, basically the introduction to viruses. And then it introduces replication of viruses, and it doesn't go into the details of that until you, I forgot about that, until you get to Chapter 8. So notice this is, in this PowerPoint, I'm going to make a jump, right, to Chapter 8 when we get to replication, which is not going to be today. Yeah, it's not going to be today. I don't think so. But that's okay. All right, so on to the next. So just basic history. Obviously, the study of viruses is the study of virology. We know viruses have been, some scientists believe they were probably the first things on this planet. But they've definitely been around a very long time. So they found evidence in mummies as far back in Egypt of having smallpox, which is a fear too, where they found some bodies preserved in the tundra. And, you know, there's obviously worry that when you're getting these bodies out of the ice, that they still could potentially be infectious. And how did scientists first figure out that there was something other than bacteria? And this was very early on. Is that they literally took, you know, plants, and these plants were diseased, and they filtered them and trapped the bacteria, but yet there was still something getting through the filter that was smaller than bacteria that was actually infectious to these plants, right? The tobacco mosaic virus is one of the ones that we're going to talk about today. And then, of course, good old Walter Reed, which is famous here, right? So it's kind of cool that they picked him for this book. I don't know that I need this. I forgot to hit my button. Too many buttons, y'all. So we have the Walter Reed. We had the Walter Reed Hospital. It's been closed down now. But you know, he was very famous for making the link between the yellow fever virus and what we call yellow fever. And that was back in 1901. And then it was transmitted by mosquitoes. That was really the most important part, is that virus was transmitted by mosquitoes. And we did have that here in Louisiana. Now it's more prevalent, even though we have vaccination for it. It's still a problem in South and Central America. This virus is very interesting. It needs to actually multiply in the mosquito for a certain amount of time before it can be transmitted to a person. So, you know, you can't bite one person, pick up the virus, and then transmit it right away to the next person, which is kind of good, and maybe other than vaccination led to one of the reasons. And, you know, once we figured out to stay away from mosquitoes, that's probably why we don't have it here. The other reason why we don't have it here, so we don't have a human population that's harboring the virus, right? Why they have problems with it in South America and Central America, anyone know? No. So if humans aren't harboring the virus, if the mosquitoes are picking up and transmitting from person to person, something else has to have the virus in it that the mosquitoes are biting and then transmitting to humans, right? No. It's an animal, for sure, other than a human. Monkeys. Monkeys are the problem. So they're not going to get rid of all the monkeys, right? And good luck vaccinating them all. It's like the problem that we run into with rabies, right? Good luck. I mean, there's no way we could vaccinate all the wild animal population against rabies, right? So we vaccinate what? Our companion animals and ourselves if you work at a veterinary clinic, right? Where your exposure is higher. we vaccinated our companion animals to actually create a buffer zone between us and the wild animal population. Because they're the ones more likely to come in contact with a wild animal and get the disease and then transmit it to us. And the scary thing about rabies, too, and viruses in general that we'll talk more about later, is we have very few antiviral drugs, right? And especially with rabies, like so few people have gotten it over the years. We don't know if the treatment that we did saved them, or what's the main thing that saves you when you get a virus? Your white blood cell, your immune system. Your immune system is what saves you when you get a viral infection. Okay. And that's why they can be quite serious. So what are viruses? Right? So we'll do a simple definition of them, but then next time we're going to really discuss the implications of this definition, right, and how we define viruses. So they're intracellular obligate parasites, which means they must get into a cell. And the good news is they're specific, right? The bad news is I hate this diagram, but I kept it in for a reason to explain it to you. I think, we're trying, if you actually read, right, the figure description on this, the purpose of this picture is to show you size relationship, okay? But what I don't like is they have the Pox virus or the herpes virus, tobacco mosaic virus, that infects the plant, right? Pox viruses, herpes viruses infect us, right? And yet they have it inside of what? They have it inside of a bacterial cell, right? And E. coli at that. These things do not infect E. coli, right? It's just to show you that they're smaller than a bacteria. And then look, there's a line here, and they're saying that like E. coli is inside a human liver cell. Yeah, no. E. coli is not it's not an intracellular bacteria. It doesn't grow inside liver cells. Okay. Again, it's just meant to show you size. Okay. The important thing is that they are small. So a bacteria, remember, like those of you guys in lab, we're talking around like 200 nanometers, right? These guys are half the size of a bacteria, or a tenth of the size. So, I mean, look at all these viruses that they packed in here in this poor E. coli, which the only one that would actually infect E. coli is the T4 bacteriophage. All the rest of them are like human viruses, or especially with the tobacco, one infects tobacco plants. Their genome, right, is typically just a few thousand to 200,000 nucleotides in length. So not a lot of DNA. I guess, they're small too, right? So you can only pack so much DNA into a virus, right? So not a lot, although they talk about some superviruses now with really huge genomes, right? So remember the wonderful thing about science, right? It's always changing. It's always changing. All right, so this objective has got a lot, and I want to clean it up, and I will, but I didn't get to it today. So lots of definitions here to go over, and I really should write them out on a slide, and I haven't done that yet, right? But it's good homework for y'all, actually. Y'all should write out these things, right? So a viron is literally one virus particle, right? Just one. When we say virus, we're usually talking about more than one, and when we say viruses, we're talking about more than one. When we're talking about a single virus, right? So just like when we talk about a single bacteria, the single name for just one virus that you're looking at is a viron. There are two major types of viruses that we talk about. We talk about naked viruses versus envelope viruses. And again, this relates to our cleaning discussion, right? Because some viruses have an envelope, Others don't, and so therefore scientists said that they were naked. Because the first ones they discovered had an envelope, right? And then they found these other ones that didn't have an envelope, and they're like, oh, they're naked, right? Not really. They're just don't have an envelope. Don't have that extra layer. So nucleocapsid, nucleo refers to nucleic acid, right? Capsid is our next definition. Capsid is the name of the protein code. So naked viruses, another name for them is a nucleocapsid, because all they are is nucleic acid and a protein code. The proteins that make up that protein code, the capsid, are called the proteimmers. And then they come together in complexes, right? Those are your capsomeres, and some of them have unique arrangements I will look at probably next time. Some of them form pentamers. So how many proteins are in that capsomere, in that collection of proteins?
So pentamer is 5.
5. Pentamer is 5. There's a reason why it's called that. And then hexameres?
6.
6, right? So 6 proteins arranged, right, in that protein code. So that same protein repeated 6 times in an arrangement of 6. And so we'll look at some pictures of these, because pictures help, right? And then I even have a handout for you guys today. And you can go home and play with it. Okay. So the capsids, the protein codes, can have different shapes. So we're going to look at those shapes. So they could be helical, and then the most common is the isohedral, and this is a 3-dimensional object, right? So that one's the hardest one for y'all to visualize, and that's why I have some origami homework for you, I guess you could say. And have you, if you want, make an isohedral. And then some of them can be even more complex than the isohedral, which we'll look at in a moment. I don't have to walk back over there. I'll remember this. Eventually I'll remember this. So remember, I mentioned in the definition they're going to have nucleic acid. And a lot of times, scientists too, when they talk about viruses, and notice this table, y'all. Do I expect you to memorize anything in this table? No. This is a reference table, right? So that we have examples to talk about, right? As it relates to these things. What's important is at the top of the slide, right? That they can have either DNA or RNA. It's a choice, y'all. They're either one or the other. And then typically when we talk about DNA, like in our cells, is it double-stranded or single-stranded?
Our cells?
Yeah, our cells. Double-stranded, right? DNA is always double-stranded in our cells. Where RNA is usually single-stranded. Yeah. But look at this. Of course, on this list, they don't have it. I need another list. Okay. But, look, still, what do you think SS stands for? The genetic material for the polio virus. Single-stranded, right? And DS for the DNA? Double-stranded. Why I just realized I don't like this chart is because guess what? Viruses can have, and we're going to see this later, double-stranded RNA, and they can have single-stranded DNA. So it really isn't either or. It can be single-stranded or double-stranded. It can be DNA or RNA. It's never both, right? It's one or the other for both of these choices. Probably should even separate them out, right? So
is the influenza, like the common flu, is it the RA? Is it RNA?
You're going to ask me that.
Yeah. That's the one that's pretty hard to get a vaccination for, right?
We're gonna talk about it.
All right.
I don't want to say it wrong.
All right.
But it's one of those ones I don't have strictly memorized, so obviously I'm not going to make you all memorize it. I should, out of the fact that y'all ask me it like every time, but I don't know, it just doesn't want to click. So I'm not a virologist, y'all. I'm an immunologist by trade, right? So although I would like to, take a graduate level class in my copious amounts of free time on viruses, I have not done that. But virologists, like one of my colleagues that I did have some really interesting conversations with until she went somewhere else, they would even refer to viruses just as he's wanting to do. Oh, yeah, this particular virus is, say, an RNA virus, and even be more specific and say single-stranded, or even use the classification system that we're going to look at later, right? I want y'all to know the basics, okay? I don't want you memorizing, right, that influenza is whatever it is, okay? Does that make sense? Right? We just use these examples because, right, when we talk about things that you've heard of, and these things stick in your brain. Okay. All right. So, at the very least, it is nucleic acid, DNA or RNA, single-stranded or double-stranded. And then it's surrounded by a protein shell, right, or coat, which makes sense to protect that nucleic acid. We call that the capsid, right? Those two things together, the protein coat with the genome, with the DNA, or RNA, notice we say genome because it could be one or the other, right? It's genetic information, is called the nucleocapsid, or we could even say, at that point, if we stop there, it's what we call naked or non-enveloped viruses. Then they can have a possible, not all of them do it, right? But then they can have a possible additional layer, right? It's like a plasma membrane, and you'll see why, around that capsid, around the protein layer. So you can see a lot of the viruses, like I said, that infected us too, a lot of them are enveloped, which is good news because a membrane can be disrupted by chemicals, right? So when you guys are reading about your chemical cleaners, a lot of the viruses, influenza, I can tell you definitely is an envelope virus, right? So cleaners that can disrupt that envelope can deactivate, or their term kill, right, the virus so that it doesn't make you sick, okay? Because it's an enveloped virus. The ones like Polio Virus that they have listed here that's non-enveloped, that's a little bit harder to disrupt because we're having to affect a protein, right? There's no envelope, there's no plasma membrane layer to interfere with. Proteins are much stronger, right? Much harder to interfere with. Any questions at this point? All right, so let's talk about those capsids, those protein coats. So when you look at the tobacco mosaic virus, this first picture, under the electron microscope, right? TM, transmission electron microscope, they look like rods, right? But in fact, if you were to look at them, right, much more closely with the more high-powered microscopes we have now, the DNA is in the center, so the protein is on the outside. All those little purple spiky things, right, are the protein subunits, right, the protomers. And they're arranged around the RNA in a helical fashion, right? So like a spiral staircase. This is pretty uniform symmetry, right? The isohedral, right, which is the more difficult one, right? When we look at this one, so it has 20 of these equilateral triangles, right? 20 what we call faces, 12 vertices, 12 points. And so the example one that they have there, the adenovirus, even has spikes coming out of some of those vertices that you can see there in the picture. So that's what I have for you for origami homework, is to build one of these. And I'll tell you how to do it. Hopefully. We'll have time today. I know we will. Okay, so then they can be much more complex. So remember, this is bacteriophage, right? These are what they look like, the viruses that infect bacteria. And so these are quite more complex. They'll have, the nucleic acid inside that capsid at the top, what is commonly referred to as the isohedral head. And then they'll have the tail portion, and those proteins arranged in that portion tend to be helical again. And then they'll have the tail fibers attached to the tail, and those are what are actually attached to the surface of the bacteria. And then it kind of lands on it, and I'll show you guys some cool things about your textbook now. They have interactive diagrams where it kind of steps through this, and you can see what's happening. Or you can have much more complex shapes. So I went ahead further in the chapter, figure 526, showed you some of the more complex morphologies that you can see. So the Pox virus is usually the most commonly talked about. Honestly, the other three I've never even heard of. But I just want to show you that they can have some pretty funky shapes. Definitely interesting, that are outside the norm. So, your favorite, right? Everyone's favorite. Hopefully you've gotten your influenza shot. I got mine, right? Flu virus, okay? So this virus is, there's your answer, RNA. And it's actually segments of RNA that are arranged around proteins in a helical fashion, but in this case, it's the RNAs on the outside of the helical proteins. It's kind of interesting. And there are several pieces of RNA, right? So notice how there's several little pieces on the inside. And it is an enveloped virus, right? So you see the envelope. And then there are spikes that come out of that envelope. And so there are two spikes that we're going to talk about again later, because these are the ones that are important, and this is what you're vaccinated against. So hemagglutinin, right, the one abbreviated HA in this diagram, and neuraminidase, NA. And as you can imagine, it ends in ASE, so what type of functionality might that protein also have? It ends enzymatic, right? It has an enzyme ability to it. Hemagglutinin, heme usually refers to what? Blood, right? Glutination, excuse me. No. It's a clumping. You clump blood. That protein can cause your blood to clump up. That's where it got its name. All right. All right. So envelope viruses. For the most part, as you saw with the influenza virus, and you'll see with the HIV virus, no matter what the genome is on the inside, the outside of the virus, when you look at the whole viron, tends to have this spherical shape, right, because of the envelope layer, right? No plasma membrane, no envelope, remember we refer to those as naked virus. But both of these, HIV and influenza, are examples of envelope viruses, right? Where they have the nucleic acid, the protein on the inside, and then it's surrounded by an envelope. That envelope itself can have protein spikes coming out of it, just like you see in this previous picture, right? So, that's the basics of structure, right? The good news for us is that bacterioviruses infect bacteria. Human viruses, for the most part, or animal viruses infect humans and animals, right? Plant viruses infect plants, right? All of this has to do with its replication cycle. The very first thing that must happen is it must attach to that cell, which means somehow it must recognize, right, the protein in the capsid coat or the protein sticking out of that envelope must recognize something on that cell and literally physically attach to it. That's the first step of viral infection. So the good news with us is if we make antibodies against it, the antibodies bind to the virus and the virus doesn't bind to our cells, and we stop them right there. They don't even get it. But of course, if they're going to replicate, the next step is they've got to get into the cell. Entry, right? Virus attaches, virus goes in. Because remember, they're obligate intracellular parasites. They have to get inside. Well, let's think about that for a second. If you're infecting a bacterial cell, what layers do you have to get through to get inside that cell? Potentially. The membrane? Cell wall. Ooh, that's tough, isn't it? And that's why, right, when we talk about each one of the different types of viruses, entry is going to be different depending on what organism you're trying to get into, right? What cell? Plant cell, animal cell, bacterial cell. What type of virus are you? Right? Are you an envelope virus? Are you a naked virus? And then once it gets into the cell, it has to come apart, right? And then the very important part is it takes over our cells, right? And it uses our cells' machinery to synthesize its viral proteins, and oops, I left off. And of course, it copies their nucleic acid. How did I miss that? I was typing too fast. And then we got to put those pieces together, right? So the nucleic acid is packaged into the protein, and if it's an envelope virus, we need to add an envelope layer, and then those prodigy, those babies, right, are going to be released from the cell. And again, depending on what type of cell, right, or virus you're talking about, that can happen differently. This is the general order, right? Attachment, entry, synthesis, assembly, release from the cell. But we're going to go through, right, in our next lecture, which is mainly, right, some of that stuff you'll have to jump to chapter 8 to get the details on. Okay, where am I on top? I'm actually doing pretty good. Okay, so, like, way better than I thought I would. Why in the past is it taking me so much more time? I don't know. I'm just getting better at it, right? Alright. Let's go ahead and start. I was going to leave this, but we'll start. So as I said, chapter 8. Okay. So stick to the cell, adhere, what I call attach, and what they call attach in the diagram. On your book, they'll use different terms sometimes, right? And then it must get into, penetrate that cell, right? And when it gets inside, it's got to uncoat, right? Separate from nucleic acid from the protein. And then we've got to synthesize, and we'll talk about that later. That can be quite difficult depending on what type of virus. So in this diagram, it's a DNA virus. That's real easy, right? Already in the cell is RNA polymerase. It can make that DNA into messenger RNA, which is read by the ribosomes, and we can make viral proteins. Bam. No problem. And we can copy DNA, right? DNA polymerase is in that cell, so we can make copies of the DNA. And then we're going to assemble, right, viral DNA, protein DNA, capsid, right? And then we're going to exit the cell. So this is for a naked virus, this diagram. They say self-assembly, right? I don't believe that. Just like in our cells, remember how we learned how our proteins have tags on them and it tells them where to go? There's probably something going on here. We just haven't gotten to that level of discovery yet. I doubt it's just together. Like magic. But right now it probably seems like magic because we can't see to that level. So how do they recognize? All right, so as I said, they have specific ranges And that all has to do with that first step of attachment. Right? They've got to recognize that cell. The crappy thing for our cells is it's usually a receptor that obviously we're using, right? So we can't like shut down that receptor or stop using that receptor because then our cells aren't going to function like they're supposed to, right? Can't really use that as, say, a treatment. But over time, we evolve and we change, right? And so do viruses. So there is actually a tribe in Africa that's won the genetic lottery as it relates to HIV. Okay, so as we'll see in this next picture, where is it? That I want to show you. Right here. Here's the virus, right? There's the protein sticking out of the envelope of that virus. Here is whatever, and I hate that they make it look like an antibody, but it's not. This is some receptor on the surface of the cell for this particular mouse. Right? This is a mouse example. And mouse hepatitis. There is a strain of mice, right, so genetically different, that have this same receptor, but do you see the receptor? They're drawing it a little bit differently here. And it's different enough that the protein for that virus can no longer bind to that receptor. So if it can't attach, can it enter into those cells? No. Okay. So for HIV, right, it attaches to a co-receptor why is it called HIV? What does HIV stand for? Human Immunodeficiency Virus. So it attacks our immune system, right? It attacks specific cells of our immune system. Anybody know what ones? T cells. More specific even. You know what type of T cell? Helper T cells is another name they're given, or CD4 T cells. And the CD4 is what's important because that's the receptor that has a co-receptor that the two bind to. And this is how HIV infects those cells. It attaches to those receptors. So the good news for a tribe in Africa is that their receptors on those cells, right, on that specific cell population that HIV targets, is genetically different. It's different enough that the virus doesn't attach.
So it makes them immune to HIV?
Yep. They're immune. So can we get that from them? If you go mate with them, maybe your children, right? Right? But that's the only way, genetically, right? We're talking about genetically, the information on how to make those receptors, theirs is changed. They basically won the genetic lottery. That virus no longer attaches to their receptor. They still have the receptor. The receptor still works. It still does everything it's supposed to do. The good news for them, though, is a virus, a very nasty virus, can no longer attach to them.
That's the reason why. That's what we don't know why it doesn't attach.
Because it's structurally it's changed, right? Because again, this is a molecular interaction that we're trying to show here, right? This is molecules attaching to molecules. If those molecules can't attach anymore, right, that very first step that needs to happen can't happen. So right at the molecular level, right, there's been a change, a significant enough of change that virus's protein cannot attach to those cells. But their T cells still work. They still do everything they're supposed to do. So and that one's even on the list. First one, CD4, right, is the receptor for HIV. So for some viruses, right, they're pretty specific as to what cells they infect. Like the Epstein-Barr virus, right, which infects B cells, which is another part of our immune system. The receptor we know is the CR2 receptor, right, that particular virus is binding to and getting into those cells. That virus, that infection is known as mononucleosis, because it'll cause the B cells to replicate uncontrollably and produce antibodies that it's not supposed to be making. That's what your B cells make. It's also sometimes referred to as the kissing disease, right? Because typically you get that in junior high or high school because that's when you start kissing. But you can also get it from just sharing a drink with someone because it's transmitted through what? Saliva, right? And that's actually, what you can exchange when you kiss somebody too. Right? So other viruses will infect multiple cells. The influenza, right? So notice that it's a receptor that apparently, right, has various functions and on lots of cells, which is why it's such an ugly virus that we will continue to talk about. Right? We'll keep talking about this one. So one of these days I'll remember it's RNA. I don't know why I don't. Okay. So ways of entry, right? So this is where I'm going to go to our book and show you guys some cool stuff from our book on how some of the figures are actually interactive. So remember, this is showing you separate things. Different viruses, right? So viruses that infect bacteria, the influenza virus, which infects us, right? Adenovirus, which infects us, but influenza virus is an envelope virus, where this one is a non-enveloped or naked virus that has spikes, right? Has these protein spikes. Where this next one, like the polio virus, notice it doesn't have any protein spikes coming out of that capsid. It's the capsid directly, those capsid proteins directly interacting with the virus. So, but what's happening in all of these? Right? It's binding to a receptor on that cell surface, right? Some protein component of that virus is binding to a receptor on the host cell, the cell it's going to infect surface. Right? That, again, the most important thing, right? Attachment. So, wrong mouse.